Okay, so we are going to finish up the last part of chapter 6. And this last portion talks about how organisms live together. And then we're going to finish up talking about the bacterial growth curve. Okay, so when we talk about ecological, ecology just kind of meaning how, different organisms interact in a particular habitat. Most microbes live in shared environments, okay, or habitats, right? You have several species of bacteria living all over your skin, in your gut, out in the environment. So normally when you have all those different species, some of them are good guys, some of them are bad guys, and then some of them are like, they're not doing you any harm. They can also be obligatory or non-obligatory. And remember, obligatory means you have to, okay? Like we are obligated to pay, the government taxes every year. So it means that sometimes organisms have to live in that particular environment or they'll die. That's obligatory. Or non-obligatory means, whatever, we can live there, but we don't have to. We can actually live in other habitats as well. So often when you have organisms living together, it's often involving nutritional interactions. And what I mean by that is, for example, if you synthesize this vitamin for me, I'll make this particular amino acid for you. And so they actually become dependent on each other for particular nutrients that they might need. So the term symbiosis is a broad term, and it simply means organisms living together. Okay, so if you have an apartment and you have a roommate, you're in a symbiotic relationship. If you're living with your family, you're in a symbiotic relationship, just meaning that you're living with other organisms. Okay, same thing is between us and the microbes that live in our body, and then also microbes that live with each other. Okay, so that's a broad term. There are 3 particular types of symbiosis. Okay, so one of these we've already talked about, which is parasitism. And parasitism, remember, is basically when one organism lives at the complete expense of another organism. So the one organism, which is the parasite, does nothing good, okay, for the organism it's living in or on. Normally we call that the host. Okay, so like a tapeworm, your intestines provide a beautiful, nutrient-rich, safe environment for the tapeworm. In return, the tapeworm sucks off all your energy and nutrients and makes you really sick. Okay, so, basically one organism suffers while one organism benefits. That's what parasitism is. On the other end of the spectrum, we have mutualism. So mutualism is when the 2 organisms need each other, but both of them benefit. So a perfect example of this is E. coli living in your intestine. Okay, so your intestine, once again, lots of nutrients going through there. It's safe, it's warm, because E. coli is a mesophile, so it's the perfect temperature. There's lots of nutrients. It's definitely getting a benefit. And what it does in return is it synthesizes vitamin K, which then you need to help your blood clot. So that's an example of mutualism. You're living together, but you both get a benefit. And then somewhere in the middle, we have commensalism, where one species gets benefits, but the other is like, whatever, you're not hurting me, you're not helping me either, but I really don't even notice you, so I don't care. So perfect example of this is, the 2 different species of Staphylococcus that live on your skin, which I talk about in the lab bacterial species lecture. So you have Staph aureus and you have Staph epi, and they're always on your skin. So your skin is a high salt environment because you're constantly sweating and then going through evaporative cooling. Those 2 species of staph need high salt. That's where they prefer to live. There's nutrients that they can get from, dead skin cells. Yay. And they don't bother you at all. The only time they may bother you is if Staph aureus happens to get into a break in your skin, such as a hangnail or a cut. Yeah, you might get an infection. But if your skin is intact, you literally don't notice them. You could care less, but they get a place to live that has nutrients and that benefits them. So that's what commensalism is. Okay, I also want to mention two other terms. Okay, so synergism is where two organisms live together. Yay, they get a benefit, but it's not obligatory. So they can live with each other or not. Okay, kind of a weird example of this. I don't know if you've ever heard of this, but there's this little species of this really small bird that will go into a hippopotamus's mouth, okay? And obviously hippos are gigantic organisms, and you'd think that if a bird flew into their mouth, they'd chomp down on it like, hey, a snack, great. But there's this one species of bird that the hippo allows to go into its mouth and actually clean its teeth. Okay, so the hippo gets his teeth cleaned. The species of bird gets a delicious meal of, plaque and gingivitis, which, makes me kind of barf in my mouth a little, but they don't have to live together, but if they happen to run into each other, yay. Okay, so that's what synergism is. But by far, the more important term that I want you to understand is antagonism. Okay, now normally when you think of antagonism, it has a bad definition. Okay, if someone is antagonistic, what that means is it's someone who likes to pick fights, who likes to argue with you. They're not a fun person to be around. So yeah, try not to be an antagonistic person because you're not going to have very many friends and people won't like you. However, in terms of bacterial species that live in and on you, antagonism is a good thing. Okay, so remember this phrase. Microbial, and specifically bacterial antagonism, is good. We want all of the species in our body and on our bodies kind of fighting against each other, right? They're fighting for nutrients, they're fighting for space. And when that happens, it prevents one species from overgrowing and kind of taking over and being able to cause an infection. Okay, so yep, we absolutely want our microbial species in and on us to be very antagonistic. And when I get into the chapter on antibiotics, we'll learn how that actually, when you take an antibiotic, it destroys that antagonism. And that's why you end up with some of these infections that we associate with taking an antibiotic. Like sometimes you might get really sick to your stomach, or you might end up with diarrhea, or females might end up with a vaginal yeast infection. And that's because the microbial antagonism has been screwed up by the antibiotic. Okay, but we'll talk about that later. But I want you just to keep in mind antagonism in terms of microbes actually works to your advantage, and it's a good thing. We'll also talk more about normal flora or normal microbiota. Okay, and we're actually going to do a lab on it down the line, which is lab 16. So, and some of you who in lab 2 decided to sample from your own skin or to sample like underneath a ring or a piercing or a watch, what you got a sample of on your agar plate was your normal flora. So it just means microbes that are usually normally on your skin, in your gut, other sites that aren't sterile, and they're there for the long term. Okay, they can certainly fluctuate in your lifetime and, with things like, hormonal changes like pregnancy, and maybe if you're sick, they change a little bit. But normally these are organisms that are pretty much in and on you the entire time. Okay, and between them, and actually between you and them, you can see all different kinds of relationships. They can be parasites. Obviously, you want to get rid of those. They can be mutualistic. They can be commensals. Okay, but normal flora means that they're pretty much in and on you, kind of permanently. They're there all the time. And remember, that's a good thing, okay? Because you wouldn't be as healthy if you didn't have that. Okay, so finally, we're going to talk about the microbial growth curve. And because we're mostly talking about bacteria, I'm going to, refer to it as the bacterial growth curve. So when bacteria grow, now remember, they are prokaryotes, right? So they don't have a nucleus. They don't have all that other stuff like organelles. They just have very basic components. So when they grow, you have kind of two different types of growth. One is, hey, I need to make new ribosomes. Maybe I need to make, an amino acid, a carbohydrate. You kind of have cells that, are just kind of going about their daily business. Okay, so that's growth. But then you also have when bacterial cells replicate and the population increases. So I know you guys have probably had, high school biology, you've taken other types of biology. I know a lot of you have taken A and P. So I'm sure you've heard of the processes of mitosis and meiosis. And those processes happen in eukaryotic cells, the ones that are larger and complex and that have a nucleus. Okay, bacteria, because they're small and simple and they are prokaryotes, reproduce by a process called binary fission. And it is way, way, way simpler than the process of mitosis. Much more simple. Okay, so this is what binary fission looks like. So if we look at what's in a prokaryote, just to refresh our memory, and actually let's look up here. So here's a typical bacterial cell, right? We know it has a cell membrane, which is shown here in tan. Most bacteria have a cell wall, which is shown in dark brown. They have cytoplasm, which is, the blue stuff, that's kind of the background that everything floats around in. They have ribosomes, which are these little guys where proteins are produced. And then you have the bacterial chromosome. So it's still a chromosome, but it's not contained within a nucleus. Okay, so for binary fission to happen, basically the cell gets bigger and longer. It reproduces its DNA, so it copies the chromosome, and it makes more stuff. It makes more ribosomes, it makes more components for the cell membrane and the cell wall. And then the cell wall and membrane just kind of grow down the middle, and boom, you're done. So, there's also no meiosis. You're not trying to reproduce sexually and mix up genes. The goal of binary fission is to make an exact copy of the original bacterial cell. So you go from having the original cell to two daughter cells. And in a typical bacterium, this will take about 20 minutes to occur. If you compare that to one of your cells, which are eukaryotic, much bigger, much more complex, one of your cells takes about 24 hours to reproduce itself. Okay, so yeah, bacteria can reproduce much, much faster. So we refer to this as the generation time or the doubling time, the time required for, a complete fission cycle, or basically to go through the process of binary fission. And so every time the bacterial population goes through that, it's going to increase the population by a factor of 2. And so eventually you're going to get to the point where it is logarithmic or exponential, meaning that that population is just going whoop, it's going and ends up basically going straight up. So, here they say the average time is about 30 to 60 minutes. First of all, don't memorize any of these numbers for a quiz or a test. I will never ask you that. If we get the average, yeah, it's about 20 minutes. And as long as the environment is favorable or good, what that means is it's going to keep going and going and going until things are not so good, okay? And that's what's going to help make up the bacterial growth curve, which I'm going to talk about. So yeah, typically about 20 minutes, and it's going to go, until things aren't good anymore. So this is a perfect example of, if you guys ever go to the doctor, let's say you have some type of infection and they think that, well, we think this is a bacterial infection, we're going to give you an antibiotic. Please don't go to Starbucks and hang out. Don't go visit a friend. Don't go home and take a nap. Go to the pharmacy right away, pick up that antibiotic and start taking it, because every 20 minutes you wait, the bacterial population is doubling and your infection is increasing really quickly. So it's important to get that antibiotic on board as soon as possible. So just keep that in mind. Okay, so what I'm going to finish up the lecture is talking about the bacterial growth curve. And it's a very predictable pattern, meaning that it happens with every single species of bacteria, and it's always the same shape. You may, because bacterial species, they're different from each other. They all have their own likes and dislikes. We talked about that earlier in this chapter where, some like a particular temperature, some like particular amount of oxygen. So the time of this may vary, but for every species, it's always going to have 4 components, and it's going to be the same general shape. Okay, so this is what we call the bacterial growth curve. So there are always 4 phases to a growth curve in bacteria, and I'm going to go through these individually. So number 1 is called the lag phase. Number 2 is the exponential phase. We also refer to this as the log phase, which is short for logarithmic. Okay, so exponential and logarithmic mean the exact same thing. Okay, then number 3, it will reach a stationary growth phase, and then finally a death phase. Okay, and obviously cells are starting to die at that point in time. Okay, so first let's talk about the lag phase. So the lag phase is where bacterial cells have just been introduced into the environment. Okay, so think of, you just got a cut in your skin and now you have some Staph aureus in there, and the bacteria is kind of maybe just kind of checking out its environment, seeing if it's a good, satisfactory environment to start reproducing. Okay, so the cells are not yet multiplying at their maximum rate. They're going to be multiplying a little bit, but not a lot. And in fact, if you had an infection, you would not be feeling any symptoms at this time. You wouldn't know to go to the doctor. And even if you had a test done or a sample, the test would probably come back negative simply because the cells are there in such a low number. Okay, so that's what the lag phase is. It's relatively short, though the time, as I mentioned, can vary from species to species, but it's basically the bacteria have just been introduced into the environment and they're kind of checking things out to see if it's a suitable environment. Okay, by far the biggest phase is what we refer to as the exponential growth phase, logarithmic, or log phase for short. Okay, so this is when the environment is perfect. The bacteria has tons of nutrients. There's no overcrowding with all other kinds of cells. Waste products are not building up. So it's basically life is good, everything is perfect, and we're going to keep growing and growing and growing as long as this environment is perfect. In industry, this is what they do when they grow up massive amounts of bacteria. They basically put them in a big vat of nutrient media, and they keep sucking out the old media and adding new fresh media. And so the bacteria are like, man, the party is, in full swing. It's awesome. And they're just going to keep growing forever as long as fresh media is added. Okay, so that's how we get things like insulin and human growth hormone that are grown up in these gigantic vats of bacteria. For example, a host such as you guys, okay, during the logarithmic phase, this is important, this is when the bacterial cells are most sensitive to, I refer to them as anti-microbial, but basically antibiotics, okay? And it's because even though the cells are growing really, really fast, it takes time to synthesize that peptidoglycan and to make an intact cell wall. So a lot of times the cells, the bacterial cells, are weaker at this time. And if you can get that antibiotic in there, you know, a lot of times you can prevent the cell wall from forming fully, and cells can be kind of popped like a balloon, okay? Kind of like what happens when you heat fry them in lab and they blow up, basically, okay? So the log phase, it's when the bacteria is growing at its maximum rate. It's when the environment is perfect, lots of nutrients, not a lot of waste products, but it's also when that bacteria is going to be the most sensitive to antibiotics as well. Oops, oh my gosh, I'm sorry. Okay, then invariably the bacteria is going to get to the point where things aren't so great. Okay, so we're running out of nutrients, waste products are starting to build up. So it's not horrible yet, but it's kind of getting there. So this is where the amount of cells that are being produced equals the number of cells that are dying. Okay, so it becomes a flat line. So, you know, if we were looking at a patient, this would be when the patient is starting to become septic, okay, because things are getting really bad and there's so much bacteria in that patient that there aren't enough nutrients and there are a lot of waste products. Okay, so this is when things are starting to get bad. And then finally, worst case scenario at the very end, the bacteria are going to start to die. And when they die, they are also going to die at an exponential rate. So that curve is going to go straight down. Okay, they're going to die at a logarithmic rate, just like they grew at a logarithmic rate. Okay, now in a patient, we would hopefully reach this by using an antibiotic, and the bacteria would die and the patient would live. That's our dream scenario. However, when a person dies from sepsis, both the bacteria and the patient are dying. So that's, obviously what we want to avoid. When we talk about things like disinfectants and antiseptics, this is our goal, okay? We want to use enough of that chemical so the microbes start dying, and when they die really, really fast at an exponential rate. Okay, so to sum this up, and this is the general shape of the bacterial growth curve, no matter what species of bacteria it is. So here's the lag phase, okay? So this is time, the x-axis is time. This is the number of cells that are increasing. So the lag phase, yep, just been introduced to the environment, reproducing a little bit, but not very much, just kind of getting used to the environment. Once again, if this was an infection in you, you wouldn't even have the slightest idea that you were sick, because you wouldn't feel any symptoms, and even a diagnostic test probably wouldn't pick up any bacteria because they're there in not very large number. Okay, this is the exponential or log phase. So you notice, man, it goes up really fast. And it happens really fast because bacteria can reproduce and go through their binary fission cycle in 20 minutes. Okay, so this is going to keep going and going and going until things are perfect. Then the stationary phase, which is a straight line or a plateau, happens when the number of cells produced equals the number of cells that are dying. And this is because nutrients are running out, things are not so good anymore. And then finally, you reach the death phase, and then I know this isn't continued here, but basically it would come straight down, very similar to the same pattern as you saw in the log phase or the exponential growth phase. Okay, so when we talk about implications of the growth curve, if you're working in industry, yep, that can be very important when you're trying to produce things or use bacteria to produce things. When we're talking about them in this class, and we're dealing mostly with stages of infection, where the patient is going to feel sick is basically in that log phase. And so the goal is, let's get that antibiotic, let's get it on board so that we can make that bacteria reach its stationary phase and then hopefully its death phase. Okay, so yeah, those are the implications for infection. Okay, that's the end of chapter 6. Thank you very much.